So when we talk about nitrogen bases, basically what they are is, so let us take these molecules such as adenine, guanine, cytosine, thymine, and uracil. So they are the names of the nitrogen bases. So if you observe what the nitrogen bases are, basically they are cyclic compounds. So cyclic compounds are any molecular structure that has a cyclic form, right? So by observing these cyclic compounds, we can come across two important groups, such as one group is called as purines, other group is called as pyrimidines. And it is very easy to recognize them and understand their structure. So let me help you with that. So why purines are called as purines? So let me take you here to this structure. So take a look here. We have two rings here, and one ring contains six atoms, and other ring contains five atoms. Okay. So look at this six atom ring. When you look at the six atom ring, what you observe is there are four nitrogen, and then you have two carbon. Okay. And then you have a five number ring where if you count it, it will be three nitrogen and then two carbon. So the number of atoms in these rings are not the same, right? It's different. So that is why the names also becomes different. So the six atom ring is called as pyrimidine ring. I'll write it here. Okay, this is called as pyrimidine ring. Okay, the name of the ring is pyrimidine ring, and any ring which contains four nitrogen and two carbon with this structure is called as a pyrimidine ring. Whereas the five number ring, which is made up of three nitrogen and two carbon, is called as imidazole ring. So since it is made up of two different types of rings, that is, one is a pyrimidine ring, other one is an imidazole ring. So this structure of nitrogen base is called as purines. Okay, so the examples of purines are adenine and guanine. Whereas when we talk about pyrimidines, you can see that they are only made up of one ring, and that is six atom containing ring. That is nothing but a pyrimidine ring. So here also you find that there are four nitrogen and two carbon atoms. All right. So that is why they are called as pyrimidines, because they are made up of pyrimidine ring. In purines, then you can understand by the name. Purines means it is a combination of two different rings. So that is why they are called as purines. Understood? So here, since there are two different number of atoms and the names of the rings are different, composition is also different. So that is why this structure is also called as heterocyclic structure. Hetero means different, cycle means it's not in the proper cyclic forms, there is a difference. So heterocyclic compounds. Understood? So purines examples is adenine and guanine. Pyrimidines examples is cytosine, thymine, and uracil. Understood? So let's go. Now, when we talk about the nitrogen bases, basically what happens is the combination of nitrogen bases and then the combination of your sugar and then the phosphate group together combines to form something called as a nucleotide. So when we talk about, let me come back here. When we talk about the structure of DNA, the DNA is basically made up of nucleotides. So one of the component that we discussed about the nucleotide is a nitrogen bases, right? So these nitrogen bases are nothing but purines and pyrimidines. Now the next compound that we're going to talk about is a sugar. So what kind of sugar is present in the DNA? It is deoxyribose sugar. So this deoxyribose sugar, so I'll write it here, deoxyribose sugar is made up of five carbon atoms. So that is why it is called as a pentose sugar. Okay, so pentose, penta 5, pentose sugar means 5 carbon containing sugar. So if you look at the structure of this ring, okay, this is a pentose sugar, that is deoxyribose. So now we just have to label this particular ring with respective carbon atoms. So let us say carbon 1, 2, 3, 4, and 5. Okay, so this is called as pentose sugar. Whenever we have a pentose sugar, we have 5 carbon atoms, and here each of these carbon atoms are very, very significant for us in further learning. So let us talk about the first carbon atom. Whenever to the first carbon atom a nitrogen base comes and attaches in the first position, that is in the first carbon atom. Okay, nitrogen base. So that becomes a nucleoside. Okay, that becomes a nucleoside. So we had a pentose sugar. To that pentose sugar, to the first carbon atom, a nitrogen base comes and attaches here at the first position. And when they come and attach, they have to form a bond, isn't it? So the bond between a sugar group and the nitrogen base is N-glycosidic linkage. N-glycosidic linkage. So an N-glycosidic linkage establishes between the first carbon atom of the ribose sugar, deoxyribose sugar, and a nitrogen base. So when the combination is made as such, it will become a nucleoside. Okay, it becomes a nucleoside. So we'll come back again, we'll understand thoroughly. So now we have the first carbon atom where you have a nitrogen base through N-glycosidic linkage. Look at the fifth carbon atom. When you look at the fifth carbon atom, a phosphate group. Usually, when we talk about the three important components that make up a nucleotide, we talk about sugar. Hold on one second. Excuse me. Yeah, we talk about sugar group, and then a nitrogenous base, and then we have phosphate group. Okay, phosphate group. All right. So when we have sugar, we talked about deoxyribose. Okay, deoxyribose. Correct. So when we talk about nitrogen base, we talk about adenine, thymine, cytosine, guanine, and uracil. And when we talk about phosphate group, we talk about H3PO4, that is phosphoric acid. Okay. So the phosphoric acid acts as a phosphate groups and comes and attaches to the fifth carbon atom. So when a phosphate group comes and attaches to the fifth carbon atom of the deoxyribose sugar, what happens here? A phosphoester linkage. Okay. A phosphoester linkage is established at the fifth carbon atom with the phosphate group. Understood? So when this happens, that is a nitrogen base plus sugar plus phosphate is equals to a nucleotide. So a nitrogen base is an adenine, let's say, and the sugar group is deoxyribose, and a phosphate group attaches, it will become adenylate acid. So adenylate acid, guanylic acid, cytidic acid, thymidinic acid, and uridylic acid. So all are the names of the nucleotides, which we simply represent it as A, T, C, G, and U. Okay. So which are like the codes. Okay. Understood. A, T, C, G, U are used to represent the nitrogen bases or nucleotides that are present in the DNA. So when we talk about the structure of DNA, did you understand till now? Are you guys following me? Are you able to understand? Okay, good. If you don't understand, let me know. I'll go even more slow and make you understand. Okay. Right. So if we talk about the DNA, structure of DNA is made up of nucleotides. Correct, no students? So these nucleotides are arranged in a very peculiar fashion. So the arrangement is the most unique thing that makes the DNA a special molecule. See here, if you observe the structure, this is one nucleotide, this is another nucleotide, this is another nucleotide. Okay. So you can see 5' to 3' is 1. Again, 5' to 3', sorry. 5' to 3' is 1. So these are one nucleotides that are arranged. If you see the arrangement, they are stacked like coins. That means there is equal distance between one nucleotide to another nucleotide. If you measure this difference and if you compare this difference and this difference, it's always the same. So that is the arrangement of DNA molecule. So when we see this arrangement, we have to talk about the structure of DNA. So when we talk about the structure of DNA, we have to just consider a very brilliant scientist, that is Watson and Crick. Okay. Watson and Crick are the scientists who suggested the double helical structure of DNA. So they proposed this model. So how they did it? And what was the history behind it? You will study in the +2 class about molecular basis. So right now, to just summarize what we are going to talk about, Watson and Crick are brilliant scientists who actually studied the X-ray crystallography of the DNA. So, and then they propose how the structure of the DNA molecule would look like. Okay. So today, apart from the regular Watson and Crick model that we study, what are the other models that we have? We have A, B, C, D, E, Z. That is A model, B model, C model, D model, E model, and Z model. So there are different models of DNA. Now, the most common type of model that we study and the most common type of DNA structure that is found in most living organisms are B model. B model of DNA, it is also called as the secondary structure of DNA, or it is also called as right-handed helical DNA. Okay, so let us study the structure of that. So when we talk about the arrangement of nucleotides, so Watson and Crick says that if you see the arrangement of the DNA, so the DNA shows that it is arranged in the helical form. And when the structure is in helical form, it should form a backbone and it should form some projections or sticks. So the backbone of the DNA that forms this helical curve is made up of a sugar group and a phosphate group. So the sugar and the phosphate makes the backbone of the DNA. So this backbone of the DNA, you can just assume that this is a ladder. Okay, this is the ladder and these are the sticks. Okay. So can you see this portion? The stick portion or the backbone portion is made up of sugar and phosphate group. Whereas what happens to the nitrogen bases? If you see the nitrogen bases, whereas it is adenine, thymine, cytosine, and guanine, they project towards inside. So they attach here and they project towards inside. So when they project towards inside like this, what happens to the opposite strand? Even the opposite strand, it starts projecting like this, correct, right? So when they project towards inside, they form hydrogen bonds. We'll talk about it. So when we look at the structure of DNA, the DNA is made up of two chains, understood? So these two chains are arranged in a helical form. So when we talk about the helical structure of DNA, the backbone of the DNA is made up of sugar and phosphate, understood? And whatever the projections that we have towards the inside, that is made up of nitrogen bases. Okay, so next one. One of the important characteristics of the DNA that gives it a proper stable structure is the base complementation. So see here, first we understood that the DNA is made up of two chains, correct, right? So these two chains. And then the bases are projecting towards inside and backbone is made up of sugar and phosphate. We just studied. So when we talk about the base pairs, so whatever the sticks that we have here toward which are projecting towards inside, so these bases, that is adenine, thymine, cytosine, and guanine, which are the nitrogen bases, so how do they pair with each other? Something they have to bond with, right? If they have to hold with the opposite strand, so always adenine pairs with thymine and thymine pairs with adenine, cytosine pairs with guanine, and guanine pairs with cytosine. That means for always A combines with T and C combines with G. So A combines with T with two hydrogen bonds. Okay, there is two hydrogen bonds, and C combines with G with three hydrogen bonds. So why is there a possibility that A can combine with G or C can combine with T? No, this doesn't happen with the structure of DNA. So this property of the DNA always going to the one particular base, that is adenine always pairs with thymine and guanine always pairs with cytosine. This is called as base complementation. So I'll write it as base complementation. Okay, they form base complementation. So when they form base complementation, so obviously that means, okay, whenever the adenine comes, there has to be thymine. So whenever guanosine comes, that means there has to be cytosine. Understood? So the bonds that are existing between them are very, very important. Remember this, that is two hydrogen bond or three hydrogen. So what happened now? We understood what happened, what is present in the backbone and what is present in the projections. Now we are understanding how two strands are held together. So the two strands are held together or stitched together by hydrogen bonds. Between what? Between the base pairs. Now gynosine and cytosine are there, so there are three hydrogen bonds here. Then thymine and adenine is there, so there is two hydrogen bonds here. Understood? So this happens between the two strands. So when we look at the bDNA, for the strand shows a particular polarity or the direction. So what kind of direction can you see? If I take one strand and if I take the alternate strand, so one strand has 5 prime to 3 prime direction and other strand has 3 prime to 5 prime direction. Understood? So now when we have each strand when I say that this strand is going from 5 prime to 3 prime, how am I able to tell this? We are talking about chemical molecules, right? So there has to be a basis for me to tell this is 5 prime and this is 3 prime. So if I take the structure of my pentose sugar that we talked about, that is deoxyribose, here we had the fifth phosphate group, correct, right? And here we had the first nitrogen base, and here we have the third carbon OH. We'll talk about this. So here we know fifth carbon and first carbon, we are quite familiar because we talked about how nucleosides are formed, how nucleotides are formed, correct, right? So if you see this nitrogen base, N here and then P here, so this P represents the phosphate end. That means when I write 5 prime here, that means the free end of this particular direction is having a phosphate, uh, you know, a phosphate group is just there, okay, at the free end. So this is the 5 prime end. Whereas I told you that in the third position, there is a OH group. So in the other free end, if there is a OH group, okay, that is seen, so that shows that it is a 3 prime end. So when I say that the direction of the strands is anti-parallel, so that means here it is OH, here it is P. See here, the P is in one direction here, and this P is in another direction. So the strands are running in opposite direction. So this is called as anti-parallel nature of the DNA strand, or polarity of the DNA strand. Understood? So the strand shows direction. Understand? So apart from the strand showing direction, and DNA is made up of two strands and the composition of the DNA and how the bonds are held. Now we will see how each of this nitrogen bases are organized. So when we talk about each strand, let us say we have one strand here and we have another strand here. So now I told you each of this DNA strand is made up of nucleotides, correct, right? It is made up of nucleotides. Now this nucleotide, if it has to be made of nucleotides, that means each nucleotide, let's say I have a set of nucleotides A, T, T, C, G, G. Okay. So these are my nucleotides. If one nucleotide and the other nucleotide has to be present there, that means they have to hold each other, right? So how are they held together? So let's see here. Okay. Let me find some space. Let me do this here. Okay. So what I do is I will draw a deoxyribose of one nucleotide and a deoxyribose of another nucleotide. Okay, this is the two structure, isn't it? So now here I have a fifth phosphate group and the third OH group. Again, I have a fifth phosphate group and a third OH group. Correct, right? So and it goes on. Now you can see here there is a third OH group and a fifth phosphate group, and they can come together. So when I have a nucleotide here and another nucleotide here, so these two are held by a 5 prime phosphate and a 3 prime OH. They both are held together by a bond called as phosphodiester phosphodiester linkage. Understood? A phosphodiester linkage establishes between a first nucleotide and the second nucleotide between a 5 prime phosphate group and a 3 prime OH group. Now we have something called as polynucleotide chain. So what is the chain called as? Poly. Let me erase this, students. 1 second. Excuse me. Yeah. hold on. Yeah, a polynucleotide chain. A polynucleotide chain means one nucleotide and another nucleotide are held together in a chain kind of a formation. So when they are held together in a chain, they are called as polynucleotide chain. Understood? So there are two polynucleotide chain. One polynucleotide chain is running from 5 prime to 3 prime. Other polynucleotide is running from 3 prime to 5 prime. Okay, this is the how the base pairs are arranged, and this is how the helix are held together. Okay, so if we talk about the BDNA or the structure of BDNA, which Watson and Crick proposed, we are talking about it, but some of the facts about the BDNA becomes more relevant when we talk about it, such as the arrangement of the base pairs. So if you look at the structure of the DNA, you can see that there are turns, right? So each turn has two kinds of a groove. One groove is called as a major groove. You can see here. So this groove is called as a major groove, and another groove is called as a minor groove. A small turn and a big turn. Understood? So this is called as a major groove and a minor groove. So that means there is a staircase like arrangement in the DNA strand. So if I start counting from the beginning of the curve and the ending of the curve, so from one turn to another turn, if I start counting, what I'll see is there are 10 base. See, there is 10 base pairs. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay, let's take till here. So there are 10 base pairs between each turn. So one turn and the other turn has 10 base pairs. So if there are 10 base pairs here, if you measure the distance between each base pair, so that is around 3.4 degree angstroms. So if you see the overall distance of each turn is around 34 degree angstroms. So what does it say? It says that the helical form of the DNA has curves or the turns, and these turns actually contains 10 base pairs. The distance between each turn, see the distance of the one turn to another turn is around 34 degree angstroms, whereas the distance between the each base pair that is present in the turn is around 3.4 degree angstroms. So now we have studied in angstroms. If you convert this into nanometer, how much it would be? It would be around 0.34 nanometer, and this would be around 3. Okay, 3.4 nanometer. Okay, so whenever it is given that whenever the distance is as in angstroms or nanometer, we have to just be aware. Understood? So this is about the turns and how many base pairs are present in each turn with respect to BDNA. Fine. So this is what the majority, I mean, like most of the concepts about what is the structure that is proposed by Watson and Crick, or what is the double helical structure of DNA, or the structure of BDNA. Okay. So some of the more important aspects of DNA, how it functions and why, what is it important in the biological system, we will study very detailedly in the coming year. So let us pause about DNA now and start with today's topic. So today's topic, we have to talk about metabolism. Okay, before we talk about metabolism, can I ask you if you have any doubts with respect to the B model of DNA? Students, can you hear me? Yes, no, no doubts. Okay, thank you. Chalo. Our next topic is metabolism. Okay, this is very interesting topic. When we talk about living organisms exhibit many of the properties that prove them that they're living. For example, if I ask you what are the characteristics of living organisms, you might tell growth, you might tell movement, response to stimuli, expression of feelings, right? So one major concept that proves that any of the living organism is in the living state, that is, metabolism. Metabolism is a very unique characteristic when it comes to living organisms. I know that one can argue that metabolism can also happen in the external environment. So how is it possible? So let's say that I take an enzymatic reaction that happens in my body. and I try to do it in a laboratory. If I do it in the laboratory, and if I have the same byproduct or the output of the experiment, that means that I have performed the same experiment that happens in my body. So if this is a biochemical reaction and we consider this as a metabolic pathway, even what I have conducted in the lab is a metabolic pathway. What sets it so different that from a non-living state that is a laboratory to a living state that is a human being? So when you ask this question, it is very simple. Now, when we look at an isolated system like one chemical reaction that is happening in a laboratory at one corner, to a human body where thousands of chemical reactions are spontaneously happening, and it is a dynamic state of a living being to metabolically balance everything. So I'll give you an example. If you talk about the state of living of a human or any other organisms, it is a constituent or a sum total of all biochemical reactions that is defining the living state of a particular being. For example, today how much energy I consumed, how much energy I have utilized, okay? So how much energy have I stored? What are the toxics or the waste that is produced during this process? And what is the pathway that I find out to safely remove them out of the system? What about the respiration? What about respiratory waste? All these reactions are happening within our body, correct, right? So if you take this living state, right? So it is a sum total of all these chemical reactions. Now you consider one of these reactions and just imagine that how much of the chemical process it takes to accomplish one particular process. If I have to move two compounds, let's say that I have higher concentration of sugar in one system and low concentration of sugar in another system. So now what has to happen is I have to go from higher concentration to lower concentration. Now when I say this, it becomes very easy for you to imagine because of course molecules move from higher concentration to lower concentration, right? So now we just moved molecule from one location to another location, which is one process of a biochemical reaction. So while we moved it from one concentration to another concentration, that is higher to lower concentration, the gradient has to establish, right? So there has to be this difference, that is, this is higher, this is lower. So that means there has to be no equilibrium, or if both the buckets are even, let's say this is also half full and this is also half full, will there be any movement in the molecules between them? It's not possible, right? So that is why an equality or the equilibrium should never come to place in a living system. Whenever there is no equilibrium, it enables the metabolism to balance between becoming equilibrium and not becoming equilibrium. I know it's a little bit complex to understand, but I'll make it more simple for you. See, when we talk about metabolism is a sum total of all biochemical reactions that happen in our body. Correct, right? So when we talk about this biochemical reactions, so one thing that defines these biochemical reactions, why this is so important topic is, see, if you see for any work that is done in your body, so it should never be at equilibrium. So why it should never be at equilibrium? Because when it is at equilibrium, there can be no work done. Okay, so that is why it is always a steadily unequilibriumic state, our body. Understood? So when we talk about these metabolic reactions, it can be anything. So basically, you can summarize these metabolic reactions into two important types. One is anabolic process, other one is a catabolic process. For example, if I have a complex material, that is the food I take, when I intake the food and I break down the food, the complex food is broken down into simpler matter. For example, the starch, when it is broken down into simple sugar, or when the carbohydrates are broken down into simple sugar, or the lipids are broken down into simple fats. Understood? So this breaking down process is called as catabolic process. So whenever we break down something, we release energy like a bomb. So that is called as catabolic process. And we use this energy and the simple compounds that is produced in our body. Then we construct the bigger things. So that construction process is called as anabolism. And that requires energy to do the process. Understood? So when we talk about, hold on, do we have it? Yeah. So when we talk about catabolic and anabolic pathways, which are the two phases of metabolism, so let us talk about catabolism first. So to understand catabolism, so let us just take an example of having food. So today I had lunch, and to lunch, let's say that I had some chapati or rice. So when I took it, it is in a very complex form. So as it goes into my digestive system, it breaks down into simpler forms using enzymes. Correct, right? Digestive enzymes are put on, and it is broken down into simpler forms. So let me just come down to polysaccharides and proteins that are present in my food, right? So polysaccharides, and then there are proteins. So these are the two things that were in my food. So now what my body did was it saw the protein and it starts breaking protein into peptide chains, and the peptide chains were broken down into amino acids. And during this process, I also release some waste. Okay, some, like for example, ammonia is released or carbon dioxide is released. Okay, so any of this thing is released during this breaking down process. Now once I have broken down, some kind of waste is accumulated, and some amount of the small particles or the monomers or the simpler particles of this entire digestion process are also there. Now, what do I do with this? During this process, when I broke down these proteins to peptide, there will be some bonds between these, peptide molecules. So when I break bonds, I will produce energy, and this energy that is produced is in the form of ATP or adenosine triphosphate, which is the currency of energy. For any chemical reaction that we have to do, we have to pay some price, and that comes by spending some energy. And our biological system spends this energy through this molecule called as adenosine triphosphate. Understood? Now I have broken down this complex protein into simpler amino acids. Now what is the use of these amino acids? So this amino acids, what I'll do is I will start arranging these amino acids one by one and start constructing the protein I want. So if I take some plant protein or animal protein, those proteins will not be useful for me. When I break those proteins into simpler amino acids and use these amino acids for the construction of the proteins I want, like human proteins, so that is called as anabolic pathways. For these reactions, we have to spend some energy. So one way we are spending energy, other way we are gaining energy. So this is the two phases of metabolism. So why do we perform metabolism, and why metabolism has to exist? See, every organism has their own demands. For example, if I have to be human, I have to construct so much of different things. For example, I have to construct this much amount of protein to make muscles, to make tissues, to make tendons, to make my brain, to construct my spinal cord, to construct my nerves. So 98% of so much of the biological activity is done by proteins. Correct, right? And rest of it, the minerals like calcium that is present in the bones, or the iron that is present in my blood. Okay. And similarly, if you take many examples of even the simple water that is present in your body, everything, it has to be accumulated by us. Correct, right? So how does this accumulation takes place? It comes from our surroundings. So we take food in the form of plant material, or we take animals, correct, right? Or if you can compare it to photosynthetic organisms, they use different compounds that are present in their environment, such as carbon dioxide, water, and sunlight for the preparation of glucose. So they are also in the same constant process of construction and destruction, okay, to be alive. And this is a very magical thing, because these living beings accumulate matter and construct this matter in such a way that it becomes bionic. Like all these inanimate objects like carbon, nitrogen, sulfur, phosphate, magnesium, calcium. So these are just minerals, but see what metabolism can achieve. So metabolism can achieve this complex machine which can construct these proteins, construct these carbohydrates, lipids, right, and manufacture cells. So all that is happening through a metabolic process. Understood? So when during the metabolic process, we come across two important outcomes. One is a primary metabolite, which is the result, like a product of metabolism. A primary metabolite and a secondary metabolite. So based on its significance on a biological system, such as it has to produce physiological growth, physiological process, okay, growth, development, and your reactions to stimuli, any physiological process you take, they have to support these functions. So if they're directly or indirectly involved in you to perform your very important activities as a, as a living organism, so they are called as primary metabolites. Examples: you need amino acids for the construction of proteins, you need sugars to give you energy, nucleic acids to store your information, for such as in the form of DNA or RNA, or conduct this process of life, lipids to give you insulation, to store your food, to construct your plasma membrane. Okay. Next one, vitamins, which act as an important cofactors for enzymatic activities to make your biochemical reactions much smoother and faster. So that is about vitamins. Coming to what are secondary metabolites. So primary metabolites, they are very, very important for the physiological functions, whereas secondary metabolites, when you talk about, so they do not directly or indirectly involve in any form of a physiological development in your body, or any of the important life functions such as reproduction, growth, development, etc. So what are these secondary metabolites? So sometimes these secondary metabolites are used as medicine or recreational things or some perfumes, okay, or toxins or drugs, okay, etc. So examples for secondary metabolites which are usually found in plants, fungi, and microbial cells are alkaloids, pigments, terpenoids, essential oils, toxins, okay, lecithins, and drugs, etc. So you can call this as the gifts of metabolism. So even though that they don't have any physiological significance, yet these are produced in the plant body or a fungal body or a microbial body, which have benefited humans or it has harmed humans in a certain way. Okay, so which humans have been consuming it or using it. Example of secondary metabolites such as pigments, carotenoids, and anthocyanins, so which gives like orange color or or the purple color for the vegetables or flowers. Next one is alkaloids. And alkaloids, we have morphine and codeine. So we know that morphine, basically what morphine does is it is used as a sedative during the surgery. Okay. So it will shut down all the receptors in that region and gives like anesthetic property or kill pain. Next one is terpenoids. Terpenoids, we have monoterpenoids and diterpenoids. And essential oils such as lemongrass or sandal oil, and toxins such as uh, abrin and ricin. So abrin and ricin are two types of poisons that is extracted through seeds. So basically it is untraceable, ricin. If you kill someone through ricin, it is quite untraceable. But getting a hand on a ricin is very, very difficult. It's not possible. It's very illegal. So it causes cardiac arrest in a patient, okay? And the person dies and you cannot trace what killed him. So that is ricin seeds. And then we have lectins, and then we have drugs such as, okay, so, and then we have polymeric substances such as rubber, gums, and cellulose. Examples are quite important when it comes to secondary metabolites. Please go through it and remember this, okay? So if a question comes like lemongrass, ricin, or cellulose, they are all what? Are they primary metabolites or secondary metabolites? So your option should be secondary metabolites. Or if they ask some of the examples through, give or choose the ones which are the secondary metabolites, you will need this list. Okay, so remember this. So when we talk about metabolism, we studied quite a things such as what is metabolism, what are the two types of metabolism, which is anabolic and catabolic processes. So what is the basis of catabolism? Why organisms do this? Because they need to grow, they need to reproduce, they need to function or defend themselves, okay, etc. For all this biological process to happen, to construct life, you need metabolism. Simple. And what are the types of metabolism and how the energy consumed or get destroyed, we understood. Next, we understand what are the outputs of metabolism, what is primary metabolites and what is secondary metabolites, and their examples. Understood. So now we have to understand little more important thing, that is when we talk about the conclusion of metabolism. I gave you a statement in the beginning. What is a living state of an organism? A living state is a non-equilibriumic steady state to be able to perform work. So this is a very important statement that you have to register in your mind. Okay. So now you will understand what I'm really talking about when I explained you for the first time. When we talk about a living state, so now I'm living, I have a proper pH balance, I have a proper body temperature, right? My body don't overreact, my body is not in fever, or not it is not shivering. I'm not overspending my energy, or I'm not underspending my energy. I am showing this constant physiological activities, so which prove that I'm in a living state. So for this living state to exist, for example, in a simple transportation of any substance, such as glucose, or exchange of sodium ions or potassium ions, okay, or removal of urea in your body. So if I have to do all these activities, some work has to be done, correct, right? So if I move some substance from point A to point B, so that is work done, correct, right? Some energy is put or some process is done. So for this, what is happened? For this to be achieved, a living state should not achieve equilibrium. Why it should not achieve equilibrium? So let me take two examples. One example is I have two bowls, as I gave you an example. One is having higher concentration, other one is having lower concentration. Now the movement of particles, if both the vessels are linked together, now you know that the flow of particles from this bowl to this bowl happens where the sugar molecules will migrate from here to here because there is a concentration difference. So without spending energy, they moved from this state, this point to this point through diffusion, right? So or osmosis. So when you talk about this movement, how did the difference get created? Only because of the existence of the difference in concentration, higher concentration to lower concentration. Only then the movement of particles were was possible. If it was in equilibrium, if here is also concentration is same, in this also concentration is same, and in this bucket also concentration is same, then will the movement of particles take place between the two systems? No, right? It will never happen. So that is why our body, any chemical reaction that is happening in any form, it will always try to achieve equilibrium. Equilibrium is the state of steadiness. There is no steady state, there is a complete pause, everything is in balance. So if this is achieved, okay, if this is achieved, then there is no way that the exchange of molecules can come. Okay. So every biochemical reaction in your body is achieving for equilibrium, but our body does not allow that. It will always put it in non-equilibrium steady state. It is not over exaggerating. Oh, this is too much here. No, it is too less here. No, it is a steady state that will enable the work to be done. So only when the non-equilibrium state you can get the work performed or the work done. That is why the living state is always in non-equilibrium steady state. Remember this, okay? Fine. So since it has such important values to maintain this homeostasis or biological balance, such as the concentration of blood in your body, the concentration of sodium or potassium in your blood, right? The water content in your blood, or if you talk about the temperature, the pH in your digestive system, all this is a very fragile balance. And metabolism is the biochemical reactions that can result in any chemical forms. Some will be acidic, some will be basic, some will be neutral, some will be polar, some will be non-polar. All this has to be in balance. So that happens through metabolism and maintains this steady state. Okay. Hello. So if we talk about any biochemical reactions, whenever we talk about biomolecules, biomolecules a topic is incomplete without talking the how biochemical reactions takes place. So we will talk about this biochemical reactions, especially the role of enzymes, how they, what is the activity of an enzyme, or how enzymatic activity depends. All these factors we will take it in the next class. All right. So we will conclude the session today. If you have any doubts, please leave a message. Okay. Students, were you able to follow? Did you understand? Yeah, perfect. Thank you so much. I'll see you tomorrow and we will solve some questions. Okay. Thank you.